Good evening, everyone. Due to the health emergency, we have been forced to adapt our conference to the online format. Today, we are going to talk about the organoids and it's important to study genetic diseases. And our speakers are Dr. Rivero and Dr. Rodriguez.
Begin with, what's an organoid? Well, organoids are 3D cellular structures which can be derived either from embryonic stem cells or induced pluripotent stem cells. They aim to resemble as much as possible real organ functionality and structure. This is why they have a main advantage when compared to other studying techniques, as they are more faithful representation of reality in vivo. This also allows us to study different metabolic situations such as hypoxia and as well as study more precise in a more precise manner pathophysiology of certain organs. On the contrary, they also have some limitations. As they have a simpler structure, they might have some diffusional limitations of oxygen and nutrient transport. And we may find genetic alterations associated to the culture depending on the technique we use.
Hi everyone. When it comes down to application, there are a lot of things that we can do with organoids. For example, since we can recreate human tissue, we can study the development and the regeneration of organs. They are also used in regenerative medicine. The goal is to one day replace damaged tissue with healthy tissue extracted from an organoid. They are also used in drug discovery and personalized medicine. We can create organoids from patient cells to create a specific treatment for each patient. And finally, another important application is the use of organoids in disease modeling. As I mentioned before, we can recreate human healthy tissue, but we can also recreate non-healthy tissue to further analyze what goes down in damaged tissue, which are the possible genetic causes, and what can be the next course of treatment.
Well, very interesting. And now I would like to ask if there is any question. Yes, Dr. Trinidad, go ahead.
Hello, I have a question. How could you create a 3D cultures?
Well, I think Dr. Rivero will answer.
Yes, so there are different ways in which that can be done. A simple method to generate 3D cultures is to create droplets from a cell suspension on the bottom of a petri dish. In each drop, cells will migrate towards the bottom of the drop and they will form an aggregate. This way, a 3D culture will be created.
Incredible. Thank you.
Thank you. Well, I hope you like it, and I hope you see you again in the next conference. Good evening.